I'm Mark Pegram. I'm the Associate Director for Clinical Research at the Stanford Comprehensive Cancer Institute and a member of the Division of Medical Oncology. MSI stands for microsatellite instability. Microsatellites are repeated sequences of DNA. Microsatellite instability comes from impaired mismatch repair of DNA. Mismatch is a type of DNA mutation where an incorrect letter is placed into the DNA genetic code. For example, a situation where a T or a C or a G is in place of an A along the DNA sequence. Now, there are proteins involved in mismatch repair that can correct these errors by forming a complex that binds to the mismatched section of DNA, excises the error, and then replaces it with the correct sequence, almost like a spell checker with an autocorrect function. Cells with this abnormally functioning mismatch repair are unable to correct these errors that occur during DNA replication, and consequently, these errors accumulate. This causes creation of novel microsatellite fragments that are detectable in laboratory assays. Microsatellite instability is most frequently associated with colon cancer, gastric cancer, endometrial cancer, ovarian, hepatobiliary, urinary tract cancer, brain tumors, and skin cancers. Furthermore, the familial cancer Lynch syndrome is caused by inherited mutations in either of the mismatch repair genes MLH1, MSH2, MSH6, or PMS2. In 2017, the FDA approved an immunotherapeutic called pembrolizumab, a PD-1 inhibitor, for patients with unresectable or metastatic microsatellite instability high or mismatch repair deficient solid tumors that have progressed following standard treatments. This indication is independent of PD-L1 expression, tissue type, or tissue tumor location. The accumulation of mutations due to mismatch repair deficient tumors renders them more immunogenic and thus responsive to immunotherapies like pembrolizumab. Russell Bonneville and Samik Rao-Chaudhuri, working in the laboratory, have reported the landscape of microsatellite instability across 39 tumor types in a paper published in JCO Precision Oncology, and they found MSI high in approximately 2% of breast cancers. MSI status is commonly reported on commercially available next-generation sequencing tumor genomic assays, such as FoundationOne. Therefore, one might occasionally encounter these uncommon MSI-high breast cancer cases on this basis, and they would also be considered as candidates for immunotherapeutic approaches with checkpoint antibodies such as pembrolizumab.